Hello, and welcome to my presentation on the Hardy-Weinberg equilibrium for allele and phenotypic frequencies. So, the Hardy-Weinberg equilibrium addresses the question of how genetic variation is maintained in populations over time. As you can see here in this graph, each of the lines represents a different genotype. So, one line represents the little a, or recessive homozygous phenotype. Another line represents the big A, or heterozygous genotype, and then the other one represents the big A, or homozygous dominant genotype. So over time in a population, if certain conditions are met, these allele frequencies will stay consistent in one generation after the other, which is kind of what this graph shows. So getting into some history, in 1908, Wilhelm Weinberg discovered his portion of the Hardy-Weinberg equilibrium, which is called the Weinberg law. So he was greatly influenced by geneticist Gregor Mendel, who had already studied population of genetics by crossing two heterozygous individuals. Even though Weinberg did not explicitly refer to Mendel in his work, he was clearly influenced by him. Then Weinberg studied the results of mating within a population over time and observed the ratio of alleles and saw that eventually they reached the same distribution. So again, this is called the Weinberg law. Meanwhile, in England, the rediscovery of Mendel's papers again influenced G.H. Hardy to discover and publish a concept that is very similar to Weinberg. These separate discoveries were then combined into the Hardy-Weinberg equilibrium that we know today. So, going into the equations, the two equations are p squared plus 2pq plus q squared equals 1. And then the second one is p plus q is equal to 1. So, going into what each of these letters mean, p is the frequency of the dominant allele. Q is the frequency of the recessive allele. P squared is the frequency of the homozygous dominant genotype. 2pq is the frequency of the heterozygous genotype. And q squared is the frequency of the homozygous dominant genotype. In order for the Hardy-Weinberg equilibrium to work, certain conditions need to be met. These conditions include no gene mutations can be present, so allele changes do not occur. The population size must be very large. There must be no migration of individuals into or out of a population, also known as gene flow. Random mating must be present. There cannot be any genetic drift, and there cannot be any natural selection. If these conditions are met, the allele frequencies in a population will then reach that Hardy-Weinberg equilibrium where they remain constant from generation to generation. So, what does this do? The Hardy-Weinberg equilibrium provides us a tool for studying genetic variation in populations, like I said before. By looking at the allele frequencies and genotype frequencies, researchers can predict patterns of inheritance, estimate the effect of genetic diseases and disorders, and observe evolutionary changes within a population. When the equilibrium is inconsistent or does not work, this can also show scientists whether evolutionary forces such as natural selection or genetic drift and genetic variation are present within populations. So the Hardy-Weinberg equilibrium is a principle that has shaped our understanding of genetic inheritance, evolution, and ecology, and it by explaining the relationship between allele and genotypic frequencies. This concept is able to show us what is going on in a population and predict the allele frequencies and ratios that will be present in the next generation. Anyways, so practice question. Sickle cell anemia is an interesting genetic disorder. Normal homozygous individuals, or S, have normal blood cells that are easily infected with the malarial parasite. Thus, many of these individuals will become very ill from the parasite, and many die. Individuals homozygous for the sickle cell trait, or lowercase s, have red blood cells that readily collapse when deoxygenated. Although malaria cannot grow in these red blood cells, individuals often die because of the genetic defect. However, individuals with the heterozygous condition, or capital S, lowercase s, have some sickling red blood cells, but generally not enough to cause death. In addition, malaria cannot survive well within these partially defective red blood cells. Thus, heterozygous individuals tend to survive better in a population where malaria is present. So, if 9% of an African population is born with a severe form of sickle cell anemia, or lowercase s, what is the percentage of the population that will become more resistant to malaria because they are heterozygous for the sickle cell gene? Okay, so to solve this, we have to go back to those original two equations, which are our p squared plus 2pq plus q squared is equal to 1, and p plus q is equal to 1. So 9% of the individuals are homozygous for the recessive allele, so that is your q squared. So that's 0.9 is equal to q squared. So to find q, we're going to take the square root of 0.9, which is 0.3, and then to find p from q, we're using that p plus q is equal to 1, so p would then be equal to 1 minus 0.3, so p is 0.7, and then 2pq, plug all those numbers in, we get 2 times 0.7 times 0.3, which is equals to 0.42. So 42% of the population are heterozygous for the sickle cell gene and therefore are resistant to malaria. So, anyways, thank you for watching my presentation, and I hope you find the Hardy-Weinberg equilibrium interesting.